We live in a world that is dynamic and in color. But why is vision testing still in black and white and static? To answer these questions, we must first understand two concepts, the biology of the human eye and the mechanics of vision testing. Acuity is the measure of how clearly we can see. Measuring visual clarity is a function of the size of the image and the viewing distance. The original visual acuity test was being able to identify the stars in the Big Dipper. The acuity endpoint is the smallest arc width image identifiable at a specific distance. Good acuity let early hunter-gatherers distinguish predators from game to enable them to eat rather than be eaten. To understand how we see, we need to realize that the eye is a biological machine. The eye is a sphere with four primary parts which determine the quality of our vision: the cornea, lens, retina, and fovea. The cornea is a dome-shaped cover at the front of the eye that protects the pupil and iris and bends light to help the eye focus. The pupil is the black opening at the center of the cornea that allows light to enter the eye. The iris is the colored circular area that surrounds the pupil and adjusts its size to control the amount of light entering through the pupil. Acuity is the measure of visual clarity. As light passes through the biological lens, the lens changes its shape to have that image focused on the fovea. The proper focus of that image is what lets you see clearly. Inside the retina and surrounding the fovea are rod-shaped photoreceptors which are sensitive only to the intensity of light, which helps our night vision. Accommodation is the process of the lens automatically changing its shape to enable that image to be focused on the back of the eye. When looking at distant images, the shape of the lens is flattened. When looking at near images, the shape of the lens becomes rounded. Changes in the alignment of the lens also affects acuity. Refraction is the process of a doctor using spherical, cylinder, and axis lenses to measure and compensate for optical errors in a patient's eye. Refraction compensates for the misaligned orientation of the lens. Cylinder compensates for the forward or backward tilt of the lens. Axis compensates for the horizontal misalignment of the lens. As light passes through the lens, it passes through a layer of nerve cells called the neuroganglia until it reaches the color-sensitive cone-shaped photoreceptor cells in the fovea in the central rear area of the retina. Those cone-shaped photoreceptor cells in the fovea specifically respond to the colors of blue, green, and red. Those cone-shaped photoreceptor cells function much like pixels of colored light in an electronic display. Each of the cone-shaped photoreceptor cells responds to a specific range of light so that the combination of those colors of blue, green, and red creates the millions of colors and shades our brain thinks it sees. The photoreceptor response to blue, green, and red light is then transmitted forward back to the neuroganglia cells, which function as a biological circuit board. The neuroganglia combines the response from 100 photoreceptors for each optic nerve fiber to create the specific color and intensity signals sent to the brain. But why do the cone-shaped photoreceptor cells in the fovea respond to the specific colors of blue, green, and red? As light passes through the lens, each color of light has a different focal depth in relation to the retina. Blue is focused in front of the fovea, green is focused on the fovea, red is focused behind the fovea. Visual acuity is regulated by the relative focal depths of the colors blue, green, and red as perceived by the color-sensitive cone-shaped photoreceptors in the fovea. The relative intensity and focal depths of the colors blue, green, and red result in the muscles controlling the lens adjusting the shape of the lens and adjusting the combined focus of the image on the fovea. This process is called chromatic triangulation. The current global standard for vision testing was created by Dr. Hermann Snellen 160 years ago using European-style letters he called optotypes for measuring acuity and refractions. That 1862 Snellen test was a major technical achievement, but it requires literacy with European-style letters. It also measures vision only in black and white. It is difficult to use with children or infants. It uses irregular stimulus targets. And it is inherently imprecise due to the stimulus gap of the letters being too large. Snellen methodology also requires patients to correctly guess the identity of 4 out of 5 irregular letters on a line to correctly determine their acuity. As a result, it frequently leads to prescription errors. Snellen vision testing consistently adds excess minus power and results in overcorrecting acuity and refractions by depleting the refresh and dynamic responsiveness of the eye's photoreceptors. This overcorrected and inconsistent Snellen testing may be a factor in the global epidemic of myopia. Myopia is the difficulty in clearly seeing distant objects. Myopia is the most frequent reason for needing vision correction and affects 22% of the world's population. The solution? Introducing the Dyop, the 21st century method of dynamically measuring vision and measuring the effects of color. A diop is a spinning segmented ring used as a visual target. A diop uses vision physiology rather than culturally dependent letters to measure acuity. The smallest diameter diop ring detected as spinning is the acuity endpoint and is much smaller than the smallest letter identifiable in a Snellen test. When using a diop for vision testing, 2 diop rings are displayed, but only one of the diop rings is spinning. The patient is asked which ring is spinning, the left ring or the right ring. If the patient doesn't detect either diop ring as spinning, then both rings are made larger until a spinning ring is detected. If either diop ring is detected as spinning, then both rings are incrementally made smaller and the spin location or spin direction is randomly switched until spinning by the smaller rings cannot be detected. Diop subacuity is any ring diameter where the spinning ring is too small for spinning to be detected. Increasing the diop diameter from a subacuity size to the next largest diameter where spinning can be detected verifies this larger size as the acuity endpoint. The acuity endpoint is the smallest diameter diop where spinning can be detected. To prevent guessing, false positives are easily detected by asking the patient which diop ring is spinning and which direction is it spinning, clockwise or counterclockwise. Guessing can be further reduced by repeating the test several times, where each time the location of the spinning ring and direction of spin are selected randomly. The result is that diop testing provides a rapid, precise, and accurate measure of acuity regardless of patient literacy, culture, or age. Dyop research has consistently shown that Snellen testing overcorrects acuity measurement by adding excess spherical minus power and may be a contributing factor in the global epidemic of myopia. A problem inherent with Snellen testing is that we don't see black. Snellen acuity measurement is due to the identification of the white gaps in the Snellen letters and not the black letters. The result is that Snellen and similar tests have excessively large and inconsistent gaps. That theoretical average Snellen stimulus gap is 1.0 arc-minute squared. Diop acuity measurement is made possible by the motion detection of the identical and consistent gaps in the spinning diop. The spinning diop gaps avoid depletion of the photoreceptor response by providing a strobic stimulus. The consistent diop stimulus gap is 0.54 arc-minute squared. The increased precision, accuracy, and efficiency of a diop is likely due to the size of the diop stimulus gap being half of the area of the Snellen stimulus gap. As a result, diop acuity testing is up to 6 times more accurate than standard Snellen testing. Diop acuity testing is up to 8 times more precise than standard Snellen testing. Diop testing is typically twice as efficient as Snellen testing. Diop acuity tests typically take only 10 to 20 seconds per eye. Diop refractions typically take only 90 to 180 seconds per eye. Diop testing has another significant advantage over Snellen testing. Diop testing can quantify the effect of color on vision, something which Snellen ignores. This is critical because not everyone sees color in the same way. Individuals with a stable near image typically have a 50% red and 45% green ratio of photoreceptors. Individuals with near vision stress typically have a 75% red and 20% green ratio of photoreceptors. Why does near vision stress exist? The eye developed about 300,000 years ago. Near vision stress enabled those individuals to have a stable distance image and survival advantage in spotting predators, game, and other threatening tribes. With the development of writing about 3,500 years ago, a stable near image became an advantage when reading letter-based words. Cultures with near vision stress tend to use pictographic-based words. Another negative effect of color perception by people with near vision stress is illustrated by a condition called dyslexia. Dyslexia is the reduced ability to read letter-based words and has little to do with reduced intelligence. Numerous forms of dyslexia seem to be associated with near vision stress. A simple and rapid diop color screening test for a stable near image versus near vision stress compares the response to a spinning blue on black diop versus a spinning green on white diop. As the identical diameter diops get smaller or you move away from the screen, which diop ring are you better able to see as spinning, the blue or the green? Being better able to see the smaller spinning green on white diop rather than the spinning blue on black diop indicates a stable near image. Being better able to see the smaller spinning blue on black diop rather than the spinning green on white diop indicates near vision stress. Near vision stress indicates that a person potentially has symptoms of dyslexia or possibly symptoms of migraines or epilepsy. Further research is ongoing. An online version of the test is at redgreenscreening.com. How well does the color screening test work? The diop color screening test with a blue on black versus a green on white spinning diop has a 90% correlation for diagnosed symptoms of dyslexia. Diop vision testing is also possible with infants as young as 14 weeks of age. Large groups may easily and rapidly use diop acuity testing by comparing the diop diameter where spinning is first detected versus the diop viewing distance. Diop testing for low vision is inherently more precise than Snellen testing because the diop test doesn't have the bloated logarithmic curve of a Snellen test. Our goal is for the diop test to become the 21st century standard for global vision testing. Welcome to the diop revolution, helping the world see clearly, one person at a time.